In this video, we will discuss about classification of enzyme with suitable examples. What is enzymes? Enzymes are biocatalyzed. They are very important. They accelerate the reaction without undergoing any change. That's why enzymes are known as biocatalyzed. What is enzyme? Biocatalyzed. We cannot imagine our life without enzyme. It is important to us. For example, hydrolases of protein we required nearly 24 to 30 hours with the help of hydrochloric acid outside body. But within the body, the hydrolysis will occur fraction of time. So it is very important. How we will define that enzyme? Biocatalyzed, along with there are colloidal in nature. Then there are thermolayable in character. Then there are protein except ribosome. Then there are specific in nature. What are the important characteristics of enzyme? Colloidal in nature, thermolayable in character, protein in nature except ribosome, and specific in nature. Next, we will discuss about that classification of enzyme. According to International Union of Biochemistry and Molecular Biology, they classified into 6 classes. The mnemonic OT Hill. So, first classes of enzyme, Oxido Reductase. These bring about the oxidation reduction reaction. In this type of reaction, one type of oxidase and simultaneous reduction of another substrate with simultaneous reduction of coenzyme. Examples: glyceraldehyde 3-phosphate dehydrogenase. It catalyzes the conversion of glyceraldehyde 3-phosphate into 1,3-bisphosphoglycerate. This is oxido-reductase reaction. Here, oxidase of glyceraldehyde 3-phosphate along with simultaneous reduction of NAD. NAD is converted into NADH plus H. Other examples of oxidoreductase reaction: lactate dehydrogenase, pyruvate dehydrogenase. Here, glyceraldehyde 3-phosphate is converted into 1,3-bisphosphoglycerate. This one is the oxidation. Then reduction reaction, NAD is converted into NADH2. Second classes of enzyme, transferase. It catalyzes the transfer of group other than hydrogen atom. Two type of transferase. First one is SGOT and SGPT. SGOT means serum glutamic oxaloacetic transaminase. SGPT means serum glutamic pyruvic transaminase. Okay, they bring about the transamination reaction. Another one is that hexokinase. Glucose is converted into glucose 6-phosphate. Here, transfer of phosphate group. The transfer of phosphate group of ATP is converted into adenosine diphosphate and inorganic phosphate. This phosphate is transferred to glucose. Here transfer of phosphate group, so we will get glucose 6-phosphate with the help of glucokinase or hexokinase enzyme. So, transferase. Here two type of reaction. First one is SGOT and SGPT. They bring transamination reaction. Second one is glucose is converted into glucose 6-phosphate. Here this adenosine triphosphate is converted into adenosine diphosphate and inorganic phosphate. That inorganic phosphate is transferred glucose into glucose 6-phosphate with the help of hexokinase enzyme. So third enzyme, hydrolases. They bring about the hydrolysis of the substrate with addition of water molecule. So all digestive enzymes are belong to these. Example, urease. They bring about the hydrolysis of component with addition of water molecule. Here hydrolysis of component with the addition of water molecule. Hydrolases. Fourth one is liases. They bring about the cleavage of component and they are responsible for the breakup of bond between carbon-carbon, carbon-oxygen, carbon-nitrogen, carbon-sulfur. Convert one component into two component without addition of water molecule. Here it need required water molecule. Here without addition of water molecule is known as liases. Example, glycolysis. Fructose 1,6-bisphosphate is converted into glyceraldehyde 3-phosphate and dihydroacetone phosphate with the help of enzyme altolase. This altolase belong to the liase group of enzyme. This one is belong to this liase group of enzyme. Here cleavage breakdown will occur. One component is converted into breakdown into two component without addition of water molecule. Fifth group of enzyme, isomerase. This glyceraldehyde 3-phosphate is converted into dihydroacetone phosphate. This is the isomerization reaction. They bring about the interconversion of isomer. One component is converted into another, but they are isomer to each other. Another example, glucose 6-phosphate is converted into fructose 6-phosphate with the help of enzyme hexoisomerase. One component is converted into another component. There are isomer to each other. Example, glyceraldehyde 3-phosphate is converted into dihydroacetone phosphate. Then glucose 6-phosphate is converted into fructose 6-phosphate. Last one is ligase. They bring about the synthetic reaction. For example, with the utilization of ATP. They brings condensation of two molecules, two component. Example, acetyl-CoA carboxylase or glutamine synthetase. Here acetyl-CoA is converted into malonyl-CoA. This reaction required ATP molecules as well as carbon dioxide. With the help of enzyme acetyl-CoA carboxylase. This reaction is important one. This enzyme is a rate limiting enzyme. So last one is ligase. It is a synthetic reaction. It required ATP energy as well as carbon dioxide. After that only acetyl-CoA is converted into malonyl-CoA with the help of that acetyl-CoA carboxylase enzyme. It is a rate limiting enzyme. So in this video we discussed what is enzyme, that classification of enzyme, oxido, reductase, transferase, hydrolase, liase, isomerase and ligase. Thanks for watching.